Dear O-level students, today you have a special kind of practical questions paper based on bio sections from grade 10 and 11. Actually, these questions are very easy questions. My target is here that not giving the questions but remind your previous knowledge on the bio section. By looking at these pictures, if you know the whole experiments, what are the problems related to that, likewise you can think all that experiments. Likewise, you go through one by one and practice this paper. Once you complete the answer into this paper, then you can listen to my discussion. Before doing the paper, if you listen to my discussion, that is useless. Don't do like that. First you answer these 20 questions and check your knowledge. Simple questions are here, right? Then after that, you can get some idea about these questions. That means all these questions based on biological practicals, right? Okay, now I'll go through very quick discussion one by one. When you get into the glucose test, we know in the glucose test, we use brown color solution, that is Benedict solution. And the next one, when you get into the test, in the glucose test, first you get the glucose test in the test tube, and after that we have to heat it. Then you can see this kind of color change: blue, green, then orange, and the brick red precipitate. I think you remember that colors, and most of you have done these experiments. And the next one, you get into another test from the grade 10 syllabus, that is Sudan 3. It's a lipid tested with the Sudan, so you can see the red globules, red color globules. Remember, Sudan 3 also red color solution, and a powder also there. And when you get into the biurate test, there is no reagent called biurate in the lab. We have to get the sodium hydroxide and copper sulfate. These are the chemicals. And once you mix with the protein, then you can see this kind of very nice purple color, right? And the next thing, when you get into the lab, we know we have the microscopes, different types of microscopes, especially light microscope and the hand lens we call simple microscope. But if you want to observe this kind of view of onion cells, definitely we have to get the compound light microscope. When you get into the electron microscope, no, in the electron microscope you can get in the detailed structure of the cell organelles. But in this kind of one you can see in the normal light microscope. Here when you get into this practical, the 7 and the 6, all these questions based on the respiration process in the organisms, right? Here we know this one. First we have to confirm all the gas are coming from outside. We have to remove the carbon dioxide. To remove the carbon dioxide from the atmosphere, we use lime water. Lime water means calcium hydroxide can easily absorb that coming carbon dioxide. So this the function here to absorbing carbon dioxide. Then the next thing here, again, if you want to get the control setup instead of the whole setup here, initially we use the organism to check the respiration. So we have to remove the animal, and instead of the animal we have to get the same size artificial one to replace that place. And the next one, again from the respiration. In the respiration we know equal amount of carbon dioxide we release and we absorb oxygen. If this kind of setup we arrange the carbon dioxide absorbed by KOH, then there's an empty space. To fill that, you can see rising of this colored water, right? And the next one again from the grade 10 one. Here, check whether you know this one, that is oxanometer. You can see the growth rate of a plant using this. But in the practically, when you are doing this one, too difficult practical, because we have a very small potted plant here. Then there should be a very smooth string. Otherwise, if the friction is coming from the pulley and the string, then there is you can't see any reflection in the indicator. And the other one, if you want to get the very more sensitive reading, then we can increase this pointer or the indicator. You can increase in the size or the length, we can get more accurate reading. And the next one, again another important section in the bio, that is photosynthesis. Photosynthesis, very simple one. Internal and external factors needed, but only one internal factor there, that is in the green plants they have a green pigment, right? That's an internal factor. Others are called external. And you get into number 11 question, again in the photosynthesis experiment. This is very important practical, and we can easily do this practical in normal, even in a normal lab. Here when you get this, we know all the steps. First we have to boil in a water, and after that you get this green leaf into the alcohol with the test tube, and after we warm the alcohol in this kind of apparatus without getting into the direct flaming. I think you know that the reason, because alcohol is highly flammable. To stop that we use this thing. But we know in the alcohol, why we select the alcohol, because alcohol is an organic solvent. In the alcohol we can dissolve this chlorophyll pigment. That's the reason, not any other reason. Likewise, we continue this practical. And after that you get this one. There's another practical regarding the photosynthesis. I think you remember that. Here one component is there, that is sodium bicarbonate. Sodium bicarbonate here, or potassium hydroxide, right? That means if this kind of is there, that's the purpose of that, means they can absorb carbon dioxide. Then we can check whether carbon dioxide is an essential factor, not for the photosynthesis, right? And when you get into this one, and I think in the previous O-level exam paper also they asked the question based on this. This is the experiment for the testing the byproduct of the photosynthesis. Very important practical. Right, in here, if we want to increase the rate, then we can add the baking powder. By adding the baking powder, you keep in this setup under very bright sunlight, we can increase the rate, right? And the other way, if you get this leaflet plants like hydrilla or some other plant with a highly large number of leaves, that means you can increase the rate. And when you get into this one, in the inspiration, our respiration, we know two steps are there, inspiration and the respiration. In the inspiration process, we have to increase the surface area, or we can increase the volume of the body, right? Here, I think you remember this model using the mega bottle and the rubber balloons. In the inspiration, we take air into the body, mean the lungs. Here these two balloons represent the lungs. I think you know that, right? Then in the inspirations, once you take gas, oxygen into the body, what will happen? We know. We can see enlarging, or we call inflating these two balloons. Not any other change in this one, because these are not live parts of the non-living matter. And the number 15. Here what's the number 15? Here when you get our nervous system, in the peripheral nervous system, we have the two kinds of things, that means sympathetic and the parasympathetics. In the sympathetic one, if we are in a fate situation like that, then what will happen? We know what will happen. From head to bottom, you can get all actions. When you get your mouth, and when you get into your eyes, in this picture also you can see what will happen to the size of the pupil. In this moment, increase in the pupil, right? Then when you get your mouth, there is no any saliva at all. Saliva inhibits. And when you think about your heartbeat rate is increasing, and your peristalsis movements all get reduced, right? Likewise, so many actions are happening in the sympathetic nervous system. So the other side, again we can get into the normal by in the parasympathetic actions. Likewise, they also help for our actions in the body. And the next one, number 16, that's an important one about the reflex arcs. Reflex arcs. In the nervous system, nerve is the structural unit, but the functional unit is there, that is reflex arc. In the reflex arcs, mainly three parts. That's where you get the impulse from outside, that there should be a receptor in the sensory organs. From them we get the sensory neuron. Through that you get into the spinal cord. From them you send your message to the effector via the motor neuron. I think all of you know that. Again you remind that What's the wave? That is we call reflex arc. That is the functional unit in this nervous system. And number 16, again other side, that is our heart, human heart, our heart. We know ventricles and the auricles are there, left side and the right side. Left side having oxygenated and deoxygenated in the right side. And the ventricle combined with arteries and atria combined with veins. Here the pulmonary side or the pulmonary artery having deoxygenated but pulmonary veins having deoxygenated blood. That's the main difference. And the aorta is the main and the largest artery in the body having the oxygenated blood and distributed throughout the body except lungs. And from the organs or other organs, then the deoxygenated blood carried from the superior and inferior vena cavas, and while from the pulmonary veins breathing from the lungs, they get the oxygenated blood into the heart in the left auricle. Likewise, you know the double circulation of human. And the number 18, when you get into the number 18, what's the number 18 here? I think you can identify this picture. That is the filtration, ultrafiltration in glomerular. In our kidney, we know that's our main excretory organ there. Then the 3 steps are happening. In the very important one is the filtering. In the filtering process, and you get into the filtrate, if you are a healthy person, there is no blood cells or the plasma cells are getting into the filtrate. That is very, very important thing. And not only that, you should know what happened to the others and whole process in the nephron. And number 19, what's the number 19 here? Here by looking at, if you can get an idea about the whole respiratory system, all the respiratory systems and the functions and the special characteristic of any respiratory surface. If there any respiratory surface there, it should be a highly vascularized, thin wall for getting the diffusion, and it should be permeable for the gases. Likewise, these things are very favorable for a respiratory surface. So, in this figure, you can see the one of alveoli. We know in the alveoli, highly vascularized, you can see oxygen. You get the oxygen from alveoli to the blood. At the same time, we diffuse carbon dioxide from blood to the alveoli because our lungs are the refinery place of our body. And you get into the number 20th, that is my last question. Here we know the very, very simple system in our body. We have so many systems. One of the important system is the digestive system. I think you know the 2 main functions are there, that is the digestion and the absorption of digested matter. Here the digestion also, I think you know, for the digestion process, so many things help for the digestion, and some are glands, some are enzymes, and some muscles all are involved in the digestion process. And when you get the associated glands in the digestive system, started from our mouth, they are the salivary glands, and after that we can see the largest gland here, that is the liver. Then not only that, you can see the pancreas, they all help and secrete the enzymes for the digestion. But not only that, in our body so many glands also there. I think you have learned that the different types of glands under the endocrine system. In that case, we know what are the different types of glands. Okay, I think I did very fast revision. It is not the real explanation of each part. I think I put forward this one not for as a lesson but as a revision for you. If you complete your whole lessons regarding the biocides, then first getting into this one, that means first before listening to my discussion, you select the answers. After that, you check my discussion and try to get 100 marks. I hope all of you can do that. And again, I hope to meet you again with another chemistry practicals, with another 20 or more than 20 questions with some other experiments. Okay, thank you. Until then, stay safe. Thank you.